Hello class, and welcome to the third lecture for chapter 12, which is all about enzyme kinetics, inhibition, and control. And this is the third video that's part of this chapter. In the first two videos, we covered reaction kinetics and enzyme inhibition. And in this third video, we are going to talk more about the control of enzyme activity. So first, before we go into any specific examples, let's start to consider how the cell is able to control reaction rates. So something that we discussed in earlier videos as part of this chapter is the availability of enzyme. And so we talked a lot about how the availability of substrate can help to speed up or slow down a reaction. And it's also true that the concentration of enzyme will also help to speed up or slow a reaction. The more molecules of enzyme that are available, the faster the reaction rate will be overall. The less enzyme that's available, the slower the reaction rate will be overall. And so this is a way that the cell can and does control reaction rates, but this is a relatively slow process. So let's consider what the cell would need to do to make more enzyme, right? It would have to use the gene that encodes the enzyme. It would have to transcribe that DNA into RNA, and it would have to translate that RNA into protein. That enzyme might need any kind of covalent post-translational or co-translational modifications. It needs to get folded, and this whole process is, first of all, a lot of work for the cell, and it's also a relatively slow process. It takes a little bit of time. And similarly, to break down enzymes, it also is going to take some amount of work and some amount of time for the cell to do. On the other hand, what cells also frequently do is they can control the activity of an enzyme. And this process of sort of turning an enzyme on or off, making it active or inactive, is a relatively fast process. And we already discussed some examples of changing enzyme activity when we talked about enzyme inhibition. And so one type of inhibition that we talked about is active site inhibition. And we talked about competitive inhibitors, which block the active site. And frequently, as part of the reaction, as part of a cell, the product of the reaction sometimes can inhibit the reaction from happening by acting as a competitive inhibitor at the active site for that enzyme. So ultimately, the enzyme carries out a reaction, and once there's sort of enough product that's been made, the product itself will help to signal to the enzyme that it's time to stop making more of that product. Enzyme activity can also be controlled through inhibition due to binding somewhere away from the active site. And this is called allosteric regulation. And so it's sort of similar to the competitive inhibition, except the binding of the inhibitor is not at the active site. It's at some other part of the enzyme. And finally, what we'll discuss more today is using modulation from covalent modification of that enzyme. And these covalent modifications of enzymes are often ultimately controlled by signaling through hormones. So let's talk about one really important example of covalent modification of enzymes to control their activity. And this covalent modification is phosphorylation, or the addition of a phosphate group to an enzyme to again make it active or inactive. So when enzymes are phosphorylated, this usually happens on a serine, threonine, or tyrosine amino acid residue. And once again, this is a really common covalent modification to help regulate enzyme activity. And so we have on the bottom here a diagram that sort of illustrates this process of enzyme phosphorylation as a whole. On the left here, we have this enzyme in red. It's the inactive form of the enzyme. And then it goes through a process, goes around the circle to the other side, where the enzyme is in the green form, the active form. And it's active because it has this phosphate group added. This enzyme has been phosphorylated. So how does the enzyme become phosphorylated? It uses the help of another enzyme. And in this case, the enzyme that adds a phosphate group that carries out phosphorylation is called a kinase enzyme, a protein kinase. So a protein kinase enzyme takes ATP from inside the cell, and there's 3 phosphate groups on ATP. So it removes one of those phosphate groups, and it uses it to phosphorylate the enzyme, right, this green enzyme, which is the substrate of this kinase enzyme. Ultimately, helping to convert ATP to ADP, using up some energy. When the enzyme is in this green active form, when it's phosphorylated, eventually the cell might want to inactivate that enzyme again, when that enzyme no longer needs to be carrying out the reaction that it performs. And so to do this, the cell has other enzymes that help to remove phosphate groups. And these enzymes are called phosphatase enzymes. So a protein phosphatase enzyme uses hydrolysis, uses water to help cut off that phosphate group from the enzyme, ultimately helping it to return to this inactive form, this red form. And I just want to mention here that in this example for this cycle, the activated enzyme is the phosphorylated one, and the inactivated one is the dephosphorylated one. But that's not always true. So that's true for a lot of enzymes, but for some enzymes, it's actually the opposite, where an enzyme is phosphorylated to inactivate it, and it's dephosphorylated to activate it. So that's just something to keep in mind. But for the example we're using here, it's going to work like this, where when the enzyme is phosphorylated, it becomes more active. So that being said, let's go into our specific example of an enzyme that's regulated by phosphorylation. And our example enzyme is called glycogen phosphorylase. And I just want to note here that this might get a little confusing nomenclature-wise, because we have this enzyme called glycogen phosphorylase. It's going to carry out a reaction that we'll talk about in a moment, but that enzyme itself, this enzyme that's called a phosphorylase, is regulated by phosphorylation, which is the addition of a phosphate group. So let's talk about one of those differences, which is what does a phosphorylase enzyme do, and how is that different from phosphorylation? And then on the next slide, we'll go over the similarities and differences between all these enzymes. So glycogen phosphorylase, right? Glycogen phosphorylase is an enzyme that helps us when the cell is in a low energy state and it needs to free up some glucose to help it to ultimately use that glucose to help make more ATP for the cell to use as energy. And so glycogen phosphorylase help catalyze this reaction using glycogen, which as you recall is this energy storage molecule. It's a large polymer consisting of a large number of glucose monomers. So we have N glucose monomers in this glucose molecule. Glycogen phosphorylase uses glycogen as its substrate. It uses an inorganic phosphate, and it helps and it uses that phosphate as an attacking group to help cut off one glucose molecule from the end of one of the branches on the glycogen. So in terms of products, we have a glycogen that's shorter by one glucose molecule, and then the other product is not just one glucose molecule alone, it's a G1P, or a glucose 1-phosphate, which is illustrated below, which is a glucose molecule but with a phosphate group on the one carbon, right? And so a phosphorylase enzyme is this enzyme that's using phosphate as the attacking group to help cut this bond between this glucose that was attached to glycogen and the rest of the glycogen molecule. And because phosphate is used as that attacking group, ultimately that phosphate ends up covalently bonded to that piece that gets cut off, this glucose 1-phosphate. And so essentially what glycogen phosphorylase does is it hydrolyzes alpha-1,4 glycosidic bonds, but it does so using phosphate as an attacking group. And by hydrolyzing these alpha-1,4 glycosidic bonds, the glycogen molecule will get shorter and shorter. You might also recall that glycogen also has branches connected by alpha-1,6 glycosidic bonds, and those are cleaved by different enzymes that we will not be discussing in this particular video. So let's get back to those names. That might be a little bit confusing. So first we have phosphorylase. This is the enzyme we just talked about, right? Glycogen phosphorylase. This enzyme helps to carry out this reaction in which a bond is cleaved using phosphate as the attacking group. Free phosphate from the cell is used to attack at a covalent bond and cut the bond. So we're using phosphate to cleave a bond, and this process is called phosphorylasis. And then this is different from our phosphatase enzymes, which we discussed a couple slides ago. Phosphatase enzymes help to catalyze the removal of a phosphate group using hydrolysis, and this process is called dephosphorylation. And finally, we have kinase enzymes. Kinase enzymes help to add phosphate groups to their substrate, but they use ATP as their source of phosphate. And this process is called phosphorylation. And once again, I want to emphasize that this process of phosphorylation using ATP as a source of phosphate groups is different from phosphorylase enzymes, which use phosphate as an attacking group to cleave bonds. So that being said, let's look more at glycogen phosphorylase and how its activity is controlled. So first of all, we have to consider that glycogen phosphorylase has a T state and an R state, sort of similar to when we discussed hemoglobin a few chapters ago. So the T state of glycogen phosphorylate is the inactive form, and in this inactive form, there's a loop that blocks the substrate from binding, substrate being glycogen, right? And the T state is stabilized by dephosphorylation. And so that brings us back to our diagram from a couple of slides ago where our enzyme is in its inactive state when it's dephosphorylated. There are also some allosteric effectors that help to stabilize the T state. On the other hand, we have the R state. The R state is the active form, and when it's in the active form, the active site is accessible. So glycogen, the substrate, is able to enter the active site in glycogen phosphorylase, and glycogen phosphorylase is able to use phosphate to help cleave bonds, right, and make glucose 1-phosphate, individual monomers. The R state is stabilized by phosphorylation. So glycogen phosphorylate is in the active form when it's phosphorylated. And the R state is also stabilized by some allosteric effectors. So let's talk about all of that using this slide as our sort of summary of all of those different factors that go into control of this glycogen phosphorylase enzyme. So let's start breaking down this diagram. So in the top row, we have the T form illustrated. The T form is shown in red, this red square, right? This is where we have this loop that's blocking the active site, and the enzyme is inactivated. On the bottom, we have the R form, which is shown in green, and this is the active form of the enzyme, which is able to bind to its substrate glycogen and start cleaving. On the other hand, we have, aside from the T form and the R form, we have these two sort of states. We have phosphorylase A and phosphorylase B. Phosphorylase A, we see here on the right, is the form that has two phosphate groups attached to it. So glycogen phosphorylase, this enzyme, is being phosphorylated by having these phosphate groups added to it. And when it's phosphorylated, it's phosphorylase A. And when it's phosphorylase A, it is in this equilibrium between the T form and the R form, which we see on the right side of this diagram. And this equilibrium is shifted more towards the R form when it's phosphorylated. So just like we saw before, when this enzyme is phosphorylated, it is more shifted towards this active state, the R form. On the other hand, we have phosphorylase B. Phosphorylase B is the dephosphorylated form of glycogen phosphorylase. It can also exist in either the T form or the R form. And for phosphorylase B, when it's dephosphorylated, this equilibrium is shifted more towards the T form. So overall, when glycogen phosphorylase is dephosphorylated, it is shifted towards the inactive T form. So now let's talk about how does phosphorylase become phosphorylated or dephosphorylated. This happens because there are other enzymes that help to add and remove phosphate groups from the glycogen phosphorylase enzyme. So to add phosphate groups, to convert glycogen phosphorylase into phosphorylase A, we have a kinase enzyme, because as you recall, kinase enzymes add phosphate groups to their substrates. So this kinase enzyme uses ATP, 2 ATPs as a source of phosphate, to add these two phosphate groups onto these different sites onto phosphorylase, making it into phosphorylase A. And ultimately, we're using energy, right? We're turning ATP into ADP. And the enzyme that does this, like I mentioned, is a kinase enzyme. It's adding phosphate. So it's called a kinase, and it's called phosphorylase kinase because the target, its substrate, is glycogen phosphorylase. So this enzyme is called phosphorylase kinase because it adds a phosphate group to its substrate, which is glycogen phosphorylase, which is itself an enzyme that we're discussing the control of. On the other hand, to convert phosphorylase A over to phosphorylase B by dephosphorylating it, we have a phosphatase enzyme. And once again, the phosphatase enzyme uses hydrolysis to remove a phosphate group. So this phosphatase enzyme has glycogen phosphorylase as a target, but it also has other proteins, other substrates as targets, right? So instead of being called phosphorylase phosphatase, sort of like our kinase enzyme was named, this phosphatase is actually called phosphoprotein phosphatase because it removes phosphate groups from a few different proteins that are phosphorylated. So overall, that is a summary of this covalent modification of the glycogen phosphorylase, making it into phosphorylase A and B, each of which are in equilibrium between the T form and the R form, where phosphorylase A's equilibrium is shifted towards the R form where it's active, and phosphorylase B's equilibrium is shifted towards the T form, which is inactive. So overall, glycogen phosphorylase, when it's phosphorylated, is active, and when it's dephosphorylated, it is inactive. So now that we've talked about these covalent modifications, now let's talk more about the allosteric effectors. So let's first consider phosphorylase B, the dephosphorylated form of glycogen phosphorylase. So we already talked about how when it's dephosphorylated, it is shifted more towards the T form, but there are also other factors that can help even more shift that equilibrium towards the T form. One of them is ATP. And so let's consider this in terms of what's going on inside the cell. So like we said before, glycogen phosphorylase, it is freeing up glucose to help the cell ultimately use that glucose to make more ATP, which would indicate that we want glycogen phosphorylase to be working when the cell is in a low energy state and it needs to make more ATP. So when there's a lot of ATP around, glycogen phosphorylase doesn't need to be freeing more glucose, right? So that it sort of makes sense then that ATP is this allosteric inhibitor that helps shift phosphorylase B even more towards the T form, the inactive form. Similarly, G6P is another allosteric effector. G6P is this sort of part of the pathway of glycolysis that helps break down glucose into through a number of reactions, ultimately helping the cell make more ATP. And so if there's a lot of G6P, which is glucose 6-phosphate, then once again, the cell does not need to free up more glucose to help make more energy. There's already plenty of glucose 6-phosphate already there in the cell that the cell can then use to help ultimately make more ATP for itself. And I just want to note that G6B, glucose 6-phosphate, is different from what we talked about before, glucose 1-phosphate. Glucose 1-phosphate is the product of the reaction that glycogen phosphorylase catalyzes when glycogen is broken down. Glucose 6-phosphate is a different product that's also glucose with 1 phosphate group on it, but that phosphate group is now on the 6th carbon, and it is more used in the glycolysis pathway. So those are two allosteric effectors that help phosphorylase B shift more towards the T form, the inactive form. There's also an allosteric effector that helps shift phosphorylase B more towards the R form, or the active form, and this allosteric effector is AMP. So AMP essentially is a signal that we are in the opposite state of when we had a lot of ATP around. So ATP was telling us there was a lot of extra energy available on the cell. AMP is the opposite. AMP is an indicator that a lot of the ATP has been all used up and there's a very low energy state in the cell, and the cell needs to make more ATP so that it can carry out its function. And so this then makes sense that AMP would help phosphorylase B, even though it is dephosphorylated. AMP will help shift it towards the R state, towards that active state. On the other side, let's consider the allosteric effector for phosphorylase A. And the allosteric effector for phosphorylase A is glucose. So just like we talked about before, when there's a lot of ATP around, when there's a lot of glucose around, that means that there's sort of plenty of energy sources available for that cell to use to help essentially ultimately make ATP, to have more energy available and help the cell carry out its function. Like we talked about before, glycogen phosphorylase, its job is to free more glucose from glycogen molecules where they're stored. If there's already plenty of glucose available in the environment within the cell, glycogen phosphorylase doesn't need to free up more glucose. So that makes sense then that so for phosphorylase A, even though it's phosphorylated, and generally that would help it shift towards the R form, when there's a lot of glucose available in the cell, that phosphorylase A is going to shift towards the T form and become inactivated. So overall, hopefully that helps you see how both these covalent modification through phosphorylation and dephosphorylation, as well as these allosteric effectors, help the cell make sure that glycogen phosphorylase is active when it needs to be active to help free glucose, to break it down, to help make more ATP. And glycogen phosphorylase can be inactive when the cell doesn't need it to be doing that. And so now you should have enough information to start answering some of the Chapter 12 Control of Enzyme Activity practice questions.